Half a day. In this video, we're going to be looking at the IB Bio version of the structure of DNA. DNA stands for deoxyribonucleic acid, and DNA is a polymer made up of a chain of nucleotides, so the monomer for DNA is nucleotide. Here's the basic structure of a DNA nucleotide. Basically the monomer within the DNA molecule. So the first thing we notice in the center, we have our deoxyribose sugar. And that is a pentose sugar, pentose standing for 5, and we can see that we have 1, 2, 3, 4, 5 carbons, and that gives us our 5 pentose sugars for 5 carbon sugar. The other part that we see is the phosphate group, and we can see the phosphate group has a central phosphorus and it's surrounded by oxygen. 2 of those oxygen molecules are negatively charged. And then the third part is our nitrogenous base, and there are 4 different nitrogenous bases in the DNA molecule, and we'll talk about those in a little bit. For ease, there's a much simpler version of the DNA nucleotide, which is represented by a circle for the phosphate group, a pentagon for the pentose sugar, which I'm coloring in blue here, and either a box or a rectangle that represents the nitrogenous base. And this is the basic representation that you would be using in order to draw the DNA molecule. So now we're going to look at this DNA double helix structure. If you recall, our DNA double helix has a structure that resembles a twisted ladder. And if we straighten out that structure, it is going to resemble a ladder. And here we have a very simplified version of the DNA molecule that is using the very simplistic abbreviations for the sugars, phosphates, and bases. Here we see one of the nucleotides, and we have our phosphate, our pentose sugar, and our nitrogenous base. So we see that this one has 1, 2, 3, 4, 5, 6 nucleotides on each side. Now, as the nucleotide is formed, the phosphate, sugar, and the base all are bonded together by covalent bonds. The hydrogen bonds are found within the structures of the bonds between the nitrogenous bases. So these are hydrogen bonds, and all of these are bonded by covalent bonds. This is important later on for replication and transcription process. If you notice, there's a 3' and a 5' orientation here. This side has 3', this side has 5'. And if you take a closer look, you should notice that this one ends with just the sugar molecule. This one has this phosphate group added to it. So here we see the phosphate, and that's called 5', and then this side here is 3'. Where do we get those numbers from? If you remember, the nitrogenous base is attached to the 1 carbon, and then we have attached to the 5 carbon is the phosphate molecule. So our deoxyribose sugar had 5 carbons, carbon number 1, 2, 3, 4, and 5. Here we had the nitrogenous base, here we have the phosphate. So this is the 3' side of the sugar molecule, and this is the 5' side of the sugar molecule, and that's how we get the orientation. This orientation is very important for the structure of DNA, and we'll talk more about that in a little bit. The DNA molecule consists of nucleotides that have one of 4 different nitrogenous bases. The nucleotide may be adenine, thymine, cytosine, or guanine, and these are the 4 nucleotides, and their various combinations are what give the DNA its particular code. The 4 nitrogenous bases in DNA are consisting of 2 pyrimidines and 2 purines. So we see cytosine and thymine are both pyrimidines, and they are single ring structures. And we have 2 purines, guanine and adenine, and those, as we can see, are both double ring structures. One of the easiest ways that I remember which type is which is we can see pyrimidine has a Y in it, cytosine has a Y, and thymine have a Y in their names. So those are the pyrimidines, cytosine and thymine. And purines I remember as being the other two that do not have the Y in it. Purines are guanine and adenine. DNA is held together by hydrogen bonds between the nitrogenous bases. And these nitrogenous bases always pair together, which means adenine always bonds with thymine. And between adenine and thymine, there are 2 hydrogen bonds. And cytosine always bonds with guanine, and there are 3 hydrogen bonds. So one way I remember it is A and T equals 2 hydrogen bonds, and C and G equals 3 hydrogen bonds. AT2, CG3. These are the complementary base pairs. A will always be bonded to T on the other side. C will always be bonded to G. And this is in DNA molecule. One of the key differences between DNA of a eukaryotic cell and DNA of a prokaryotic cell is the association of eukaryotic cells with histone proteins. In prokaryotic cells, they're much simpler, and we refer to those as being naked DNA. And the reason we call it naked is because there's no relationship with the histone proteins. And prokaryotic DNA is simply DNA as we just described it. In the eukaryotic cell, however, the DNA is associated with histones, and we refer to this relationship as a nucleosome. And a nucleosome is going to consist of a central core of 8 histones, which are proteins, and the DNA coils around these proteins, these 8 proteins, or the octamer, and it consists of 2 copies of 4 different types of histones. And then there's a short linker DNA that goes on to bind with the next, or connect with the next nucleosome. And this H1 histone helps to bind with the next histone molecule, or to bond with the core particle that's there. The association with this histone basically contributes to the DNA being able to supercoil and pack itself. Eukaryotic cells notoriously have a lot more DNA per cell than the prokaryotic ones, and these nucleosomes help to organize the DNA so that it doesn't get jumbled up. Basically, you can think of it as a big yarn. If I just handed you yarn, just plain, you would probably end up getting it all tangled up in knots. But if I handed you some yarn and I put it around a cardboard or something like that, then you would be able to find it's very neat, and you can find the end and everything works out well. The same kind of thing goes with DNA. These histones and formation of the nucleosome help to keep everything in order and everything in a place, so the least amount of damage is done to the DNA molecule, because we know how important that is for our survival. The discovery of DNA, the structure of DNA, was a long process that spanned many years. And each scientist began to discover different things that led to the final discovery, in which James Watson and Francis Crick actually put together the final pattern and the model that we now use as the DNA model. But prior to Watson and Crick, there was Maurice Wilkins and Rosalind Franklin, who were the first to be able to discover the structure in the form of an X-ray diffraction image. This was a key piece of evidence that allowed Watson and Crick to be able to figure out the patterning of the DNA molecule. Morris Wilkins, Watson, and Crick were all awarded the Nobel Prize. Unfortunately, Rosalind Franklin had passed away due to cancer, and so she was unable to earn the Nobel, as it is not awarded to someone who has passed away. Erwin Chargaff was very important for his discovery of the base pair makeup. Avery McLeod and McCarty were important in that they discovered that DNA was the material of genes and chromosomes. William Atsbury discovered X-ray diffraction patterns. Frederick Griffith was able to isolate some information based on his work with bacteria into mice. And it goes back even to Gregor Mendel, who first began some of the work with looking at hereditary patterns. I'm hoping you might be able to stop the video and maybe look at this particular slide. I'm not really sure until I get this going whether or not it's going to be fully visible or not. But if not, you can refer to both of these websites here on the bottom. I actually got this from a Pinterest, but you can also see that there is this website here that you may also be able to find it at. And I hope this was great, and have a great day, and thank you.